Okay, so today we're going to be continuing with these physical methods of control of microbial growth. One of it is the use of radiation. We saw already ionizing radiation. Now we're going to move on into non-ionizing radiation. So non-ionizing radiation, it means like you won't create ions and radicals of these ions to destroy molecules. But you can use a sort of radiation that affects the DNA molecules by creating thymine dimers, like in the case of UV light. And UV light is germicidal at a specific wavelength, which is 260 nanometers. And then in the hospitals, nurseries, operating rooms, cafeteria, they use a UV lamp, UVC lamp, that it's defined as UVC because it is within this spectrum. And then in the labs, we also use these lamps. And as well, some of these chambers, they might have these lamps, the hoods in the top, that once you stop working with microbes, you close this lid from these hoods where you were working at, and then you turn on the UV lamp, or even before you start working, and that will act as a germicide. So whenever someone uses this type of light lamps, UV light, you will have to protect your eyes and your skin, because same as for the microbes where you create these thymine dimers, for our cells you also create thymine dimers, and those are mutations in the DNA that we know that are not good, because of course you know that adenine is supposed to base pairs with thymine. So this can create cancer if you don't use this protection. And now we have blue light, which is 470 nanometers, that kills a wide range of bacteria, so it's bactericidal as well. And this is due because it forms singlets of oxygen. So an oxygen molecule that is capable of destroying these microbes. And then microwaves, they are not bactericidals itself, because the wavelength is very long. However, of course, if you heat food there or water, the water will boil, that will be germicidal indirectly. But it is not, of course, the preferred method to kill microbes, to control the growth of microbes. So now, the next form of methods of, this is the actual graph showing you the spectrum of light that we have. So we have visible light between 400 nanometers to 750, going from wavelength increase in length, so the waves in here are close to each other. But as you move here in this direction, the wavelength increases, so it becomes wider. But the energy decreases because these type of wavelengths have less penetration than the ones within this range. Now, our visible light, of course, will be ranging from purple color up to red, according to this wavelength of light. And then under 400, we have UV light. And then the spectrum to be bactericidal will have to be the wavelength between 200 up to 295. Typical, the UVC lamps are 260 nanometers in diameter. And then we have tanning, which is between 280 and 330. And the visible light of our sunlight, the UV, not visible, the UV light from our sunlight will be between 295 and 400. Yes, there can be some microbes that are affected for a greater wavelength of light. However, not all the microbes will be affected. Otherwise, with the sunlight, we will kill it. So, in a way, the microbe has to be weakened in a form to be able to be killed by a greater wavelength of light, but that's not typical the case. So now we're going to see factors related to effective disinfection. You already saw part of it, and we're going to see how you can test those. So factors that affects the disinfection will be concentration of the disinfectant. In this case, we are talking about chemical disinfectants. The organic matter that can be found in the surface that you want to disinfect. pH of this environment where the bacteria is found or the microbes. Temperature, if it's too high, too cold, or a right temperature. And then the time of exposure. One of the reasons why people who wash their dishes in the dishwashing machine, why they get rid of this excess of food from the plate, of course, is to allow these disinfectants to basically heat the area, like the plate or the utensil that you want to disinfect, in order for this action to take place. Then in this case, you're getting rid of organic matter. You might have run out of disinfectant, and then you want to perhaps do your floor, like clean your floor, but if you over dilute this disinfectant, and then try to mop all the floor with this disinfectant, well, it won't work. Okay. And then temperature, typically most of the disinfectants that we work with, they are effective at room temperature. pH, it depends on the chemical. Some chemicals, they work around neutral pH, some may be working an acidic pH. And then the time of exposure also varies. Sometimes we may spray countertop and then immediately wipe away. But if you read the instructions, perhaps it tells you, well, it has to be sitting there for 5-10 minutes. And then it still can be effective if you wipe it immediately after you spray it. But the longer that it stays there, the more effective it is, especially if you have a lot of contamination of that area. Now, one way to test these disinfectants is by using the dilution test, in which you have, in some cases, metal cylinders that are dipped into a test bacteria. Then you dry these metal cylinders, and then you put those dried metal cylinders with the bacteria in a disinfectant for 10 minutes at 20 degrees Celsius. This will lead kind of a good amount of exposure for this disinfectant to act on these bacteria that was coating these metal cylinders. And then after this, you transfer your metal cylinder into a test tube with media, with broth. And then you incubate these for 24 hours, and then you see if you have had growth of bacteria. How do you know? Well, by the opacity of this media. If your media become opaque, that means that the bacteria survived in this after this treatment with disinfectant. And there can be some variations to test this efficacy, to test, for instance, for the presence of endospores, viruses, and fungi, not only with the disinfectant, but by the way you will coat these metal cylinders or whatever product you use, like what device you use to dip into these plates. Now, another method that it is useful for testing the efficacy of disinfectants, it is this diffusion method. In this disc diffusion method, what you use is filter paper in some cases, or specialized disc that has a specific disinfectant. So it's a filter paper soaked with this chemical disinfectant, and then you grow these bacteria in your Petri plate, or this microorganism in your Petri plate, in a closed lawn. And then after this, you put your disinfectants on top, grow them together in the incubator. Well, incubate these two together in 24 hours, and then read how much inhibition of growth you have around this disc. You will have to have a standard to know what is the actual efficacy. So you have to control for how much bacteria you have added there, how much time you have dipped your disc into the disinfectant, having the same concentration, and using the same incubation period. So that will make it more readable and more likely to give you consistent results. So here we have in this view 3 Petri plates, view from the top. And then these Petri plates have growth of bacteria. In this case, you can see this yellowish or beige color. So this is Staph aureus, this is E. coli, and this is Pseudomonas aeruginosa. And then you have 4 discs that were previously soaked in these disinfectants. On the top we have chlorine, on the bottom we have quaternary ammonium, on the left we have hexachlorophene, on the right we have ophenylphenol, 4 different types of disinfectants. And then the the clear halo around the disc represents disinfection efficacy or growth inhibition. So we have on the left Staph aureus, this is a gram-positive. In the middle we have E. coli, which is a gram-negative, and on the right we have Pseudomonas aeruginosa, which is a gram-negative. According to what you see there, what bacteria is the most resistant to disinfectants? Pseudomonas. And then which one will be the less resistant? Staph aureus. And then out of the 4 disinfectants, which one is the most effective? Chlorine. Which is the second most effective? Quaternary ammonium. Third? Ophenylphenol? Yeah. Or phenol? Phenol. Phenol. And then the last one will be? Hexachlorophene. So these 2 bacteria are kind of some of the most feared bacteria in the hospitals when it's come to contaminating wounds, catheters, etc. So Staph aureus and Pseudomonas aeruginosa. E. coli, it's part of this, of course, bacteria is part of the normal flora. However, certain strains, like E. coli or 15787, causes a lot of damage in humans. Causes foodborne outbreaks. So now this E. coli is not as resistant to antibiotics as is Staph aureus and Pseudomonas. Now, these ones, of course, are not antibiotics, are disinfectants. And then we know that some bacteria transfer genes in their plasmid, or from their plasmid into other bacteria that makes them resistant to antibiotics and disinfectants. Now, let's say that none of these have any disinfectant resistant genes. Naturally, there is a reason why Pseudomonas aeruginosa and E. coli are more resistant to these stuff than these S. aureus, to disinfectants than these S. aureus. Do you remember what is the difference between gram-negatives and gram-positives that is more than likely to make them more resistant to disinfectants? What do they have in their cell wall or around their cell wall that makes them more resistant? No. So generally speaking, you can tell, well, these gram-negative are more resistant to disinfectants than these gram-positive. Which one has a thicker wall, gram-positive or gram-negative? Gram-positives, they have a thicker wall. But then you can start thinking about, well, this one has a thicker wall. Why it's less resistant to disinfectants than these ones that have thinner wall? What does the gram-negatives have? Two plasma membranes, and in between the two plasma membranes they have the periplasmic space, right? What does this periplasmic space have? They have enzymes, right? So it's like a mechanism of defense. Well, I don't have a cell wall that is as thick as the gram-positives, but I have two plasma membranes, and there I hold my weapons. I have my my enzymes that helps to destroy these disinfectants. So then, out of these two gram-negatives, this one developed more resistance, Pseudomonas aeruginosa, with enzymes that help them to thrive better in harsher environments than E. coli, because E. coli is a commensal. So basically it lives in our intestines and intestines of animals. So they have milder genes. And that's why this one, in a way, is less resistant than this. Now, disinfectants have a specific use. So yes, chlorine, it is more effective to kill all these 3 bacteria than the rest. However, chlorine, it cannot be used in most inert areas because it is very corrosive. If you use it in wood, it's going to corrode wood. Use it in the countertops, eventually it will destroy them. So you cannot use chlorine everywhere. Now, for hexachlorophene, well, in this case, it is only good for Staph aureus, but not to these other two. So this won't be like a good choice when it comes to not disinfecting something that potentially you don't know if it's a gram positive or if it's a gram negative. Phenylphenol, it is less effective than quaternary ammonium, so this can be the second choice for disinfecting a surface area that is inert, of course. And then quaternary ammonium, it is one of these disinfectants that is highly used in hospitals to disinfect surfaces. However, again, Pseudomonas aeruginosa, it is one of the most feared bacteria because it has developed a lot of resistance, not only to antibiotics when it comes to infecting the wounds of patients, but also sometimes it has been found contaminating these jars with disinfectants in hospitals. So, and then Staph aureus, we have certain strains, not all, like MRSA, that is also resistant to a lot of antibiotics. And then, when it comes to disinfectants, it's kind of susceptible, so it's easy to get rid of. But you have to have certain precautions, of course, to not carry it with you. Like if you're disinfecting an area, and you're using disinfectants, you have to wear gloves. Not only to protect your skin from the secondary effects of these disinfectants, but also, what if you're disinfecting an area and then there is a bacteria that it is resistant to this disinfectant, and then you carry it with you in your hands. So be cautious. Okay, so check your understanding questions. If you wanted to disinfect a surface contaminated by vomit and a surface contaminated by a sneeze, why would your choice of disinfectant make a difference? Yes, you have to choose as well which one is virucidal or which one is not virucidal. And then, of course, let's say that, if in the sneeze, if the person has viruses, and then in the vomit, let's say that you have bacteria. In this case, you will be using a bactericidal versus a virucidal. You never know. One thing for sure is that typically when someone sneezes, the organic material, the mucus is less than when someone vomits. In vomit, you have more organic material. Let's say that in both sneeze and vomit, you have bacteria or you have viruses. Someone can have an infection with a virus and can have vomit, of course. But let's say that in this case it's either bacteria or viruses, and you choose bactericidal or virucidal according to what you believe that vomit or the sneeze have. Since you have more material coming from vomit than from sneeze, would you choose a stronger disinfectant for vomit or not? Like with more concentration? Yes, and what you're supposed to do? Remove the organic material, remove the vomit. And then, which is more likely to be used in medical clinical lab for testing efficacy of disinfectants? Dilution test or this diffusion test? And why? Why? That's correct, but why?
Because I think they can tell you a lot about whether it's been positive or negative, depending on like the disinfectants.
that can be part, like you can tell whether it's gram positive or gram negative.
I think also it could be a little bit quicker than most.
Yeah, it's quicker. Very good. Okay, now we're going to see in this part methods of action of different disinfectants, including halogens, which is chlorine, one of it. We're going to see surface active agents like soap. And then we're going to see other chemical disinfectants like gaseous chemosterilants. So one of these chemical disinfectants are the phenol and phenolics. So these phenol and phenolics were used first by Joseph Lister, and then he was a surgeon during the 1800s. There was a lot of mortality for surgeons around that time because they didn't disinfect the instruments nor the wounds that they were, nor the skin of the patients. And then whenever they were making the incision with the scalpel, they carried deeply the bacteria into the body of the person, and then the person ended up with infections and dying. And then remember, around that time, there were no antibiotics as far as we know, so someone either died on the track with the surgery or they die of the disinfection, of the infection. So mode of action of the phenol and the phenolics is make holes or pores in the plasma membrane. That causes leakage of the cytoplasm, and then the bacteria will die of plasmolysis. One important thing about these phenol and phenolics is that they remain active in the presence of organic matter. So these are good disinfectants because if someone coughs or sneezes and then you spray it there, you will still have, of course, the mucus there, but it will start killing this microorganism. So example, Lysol spray has this O-phenylphenol. And then it has reduced irritation and increased effectiveness. So here we have phenol ring, and then here we have O-phenylphenol. So this is the active ingredient of Lysol. Bisphenols has two phenol groups connected by a bridge, and similar as it's part of the phenolics, it is disruptor of the plasma membrane. Examples will be hexachlorophene, that it is used in some disinfectants that can be contaminating wounds, so it can control the growth of staph and streptococcus. Triclosan, it is another bisphenol, and it is very, very effective as an antimicrobial. It used to be part of some antimicrobial soaps, toothpaste, mouthwashes, kitchen plastics, but it was banned in 2016. It says there with the exception of toothpaste. It's not there anymore, guys. Colgate Total used to have triclosan. It was very good. Yes, I mean, they get rid of it because it created a lot of antimicrobial resistance. So, I mean, once in a while, there will be this cycle. Like, I don't know, I'm hoping that it will come back, but not in a while, because when the antimicrobial resistance, I mean, all this triclosan that we use ended up in the oceans and then in the rivers. So it kills a lot of these species as well because of that toxicity, but also it make a lot of bacteria to thrive that were antibiotic resistant. And then, I mean, we of course are constantly interacting with the environment, and then that made it for us to carry part of this antimicrobial resistant bacteria. Triclosan, however, it is used in some plastics still, not only for kitchen, but also for clothing, and also silver treated fabric that you maybe have heard about, like antimicrobial underwear and all that. So, and then some of these coats for the scrubs, they are coated with this. So here we have a couple of bisphenols, hexachlorophene, so two phenyl rings linked by this CH2 group. And then here we have triclosan, has similar two phenyl rings. And why it's called triclosan? Because you have 1, 2, 3 chlorine groups attached as side groups. B1-8s are effective against gram-positive and gram-negative bacteria and envelope viruses. It disrupts plasma membranes. Envelope viruses, they have this envelope that is coming from the host cell, so it's made of lipids. And then, so it's a plasma membrane, so then you will have damage to this. Examples will be chlorhexidine. Chlorhexidine is a very effective disinfectant. It is used as a surgical hand scrub, and as well, it is put in the skin of the patients before surgery. It is a little bit irritant to the skin, so I mean, you have to control the concentration, and then you have to remove excess from the skin to prevent dermatitis. We have another example, alexidine, that works faster than chlorhexidine. Essential oils. These are extracts of oils that exist in some plants, like peppermint, pine needles, and then orange peels. They have been used for centuries as part of traditional medicine or helping to preserve food. Some of these, they have phenolics naturally and terpenes, like in the case of pine oil. It has been tested that they have efficacy against gram-positive bacteria and against some viruses. But when it comes to the viruses, the efficacy hasn't been studied really well. Another example will be for these essential oils, tea tree oil. And just as pine oil, it's a broad spectrum, so it can kill gram-positive bacteria, gram-negative, and fungi. So here we have a couple of examples of the molecules found in essential oils. We have carbacrol, which is found in oregano, and limonene, that it is found in oranges. Kind of crazy, but it makes sense that some of these plants, they have their own antibacterials, right? So they protect themselves from whatever is found in nature. Halogens, we have a couple of examples there, iodine and chlorine. Iodine prevents the synthesis of proteins and also damages the plasma membrane. You can find it as a mixture with alcohol, in which you call it tincture, or If it is combined with organic molecules, you call it an iodophore. Example will be betadine, which is povidone iodide. This is used as a skin antiseptic. If someone has a scratch in their skin or has a wound, they can apply that and clean it. And sometimes can be used also to treat contaminated water. This one is also used for cleaning the skin of the person before the person has a surgery. However, some people might be allergic to iodine. So in this case, you have to ask and then do not apply it. And of course, if you don't know if you're allergic to, you apply that and then you develop a rash. That means that potentially you are allergic to. How do you know? Second time you apply it even without having any wound, and then you develop an allergic reaction, that will be your answer. This type of chemicals don't, like iodide, and I'm telling you this because you might think, wow, she's crazy. Why she's telling me, like, if I'm allergic, potentially allergic to something, why should I apply it again? Well, remember, there are some molecules that can develop a strong allergic reaction in some patients, leading them to death, but this is not one of the ones. It just leads to dermatitis. And then chlorine. It's an oxidizing agent, so it oxidizes proteins. It shuts down these proteins, these enzymes. Bleach, it is this chlorine that when you combine with water, you create these radicals, which is hypochloric acid. And then we have commercially available, not homemade, never do that. We have chloramine, which is a combination of chlorine and ammonia. You may have heard of it, but if you have, let me know. What will happen if someone combines chlorine and ammonia at home? The person would pass, but why? Pass momentaneously or get intoxicated, right? But why?
Does it create like a gas maybe?
It creates a gas. It creates a gas. And then chlorine, it is the main choice for disinfect municipal water. Now, alcohols, they denature proteins and dissolve lipids. So, alcohol, it is one of these agents that can be used as a disinfectant and also an antiseptic. So you can use it in your skin, or also you can use it in their surfaces. And then it doesn't have an effect for killing endospores or non-envelope viruses. So when it comes to envelope viruses, again, they carry the plasma membrane as they exit the host cells, and then that protects them. But since this membrane is made out of lipids, if you use an agent like alcohol, chlorhexidine, or a detergent that dissolves these lipids, that renders these viruses non-functional. Envelope viruses are more fragile than non-enveloped. Those of you who weren't cold today, you're not wearing an envelope because you don't feel cold. Take away this envelope from me and I will be like. I'm more fragile than you. So we have certain types of alcohols like ethanol and isopropanol, and they require water to be effective. Alcohol-based sanitizers, they have a concentration of 62%. The other 38 will be water. And then they are effective against most bacteria. And again, they don't affect endospores or non-envelope viruses. And one of these bacteria that is not affected by this alcohol will be Clostridium difficile or noroviruses. And norovirus is one of the main causes of diarrheal illnesses in the United States in outbreaks. So here this table is showing you the different concentrations of ethanol and the efficacy against Strep pyogenes, the bacteria that causes strep throat. And then this concentration, it is tested at different times of exposure in seconds, 10 up to 50. If you look at the legend, G means growth and G means no growth. So the bacteria either grow or didn't grow. In 100% ethanol concentration, the bacteria grow all the time, and then same for 40%.
Why would bacteria grow 100%?
Well, you need water to help to create this gradient between the cytoplasm of the bacteria and the outside so that you can damage this plasma membrane. So you need to have water. So I guess that's why bartenders sometimes they can dilute your drinks. They're trying to, well, let me give you a bactericidal dose of your drink. As long as they don't go very diluted. Well, it depends on how many you had before. You won't know. Anyway, so at least the minimum concentration that you can have for here to be efficacious all the time will be 60%. So between 60 to 95%, you will prevent these bacteria from well, you will kill bacteria. It's actually bactericidal. Let's call it that way. And then 50% up to 20 seconds is bactericidal, but bacteriostatic. But after 30 seconds will become bactericidal. So in this case, you call it bacteriostatic. So if you withdraw that 50% concentration from wherever you are disinfecting, then you have bacterial growth. But then if you continue soaking it in this concentration, after 30 seconds, you will start killing the bacteria.
So when they're making the disinfectants, how do they decide the concentration? Because 95% to 60% all have the same, like across the table.
Typically, for instance, manufacturers, they sell this 70% ethanol over the counter. They don't want to go so high because in very high concentrations, and we're talking about the use in skin. I mean, if you use it here in the countertop, it doesn't affect so much if it's high concentration, as long as it's something that you can put it on, of course. But when it comes to the skin, if you put a high concentration of alcohol, you dehydrate so much the skin, and that will be irritant. And then, why they don't go to 60? Well, because there is a potential that whatever you are disinfecting might be kind of highly populated with bacteria, and they don't want to go with this because if it's highly populated with bacteria, maybe it won't be as effective. And now I have seen that they sell a lot like the 91% isopropanol right now. I guess like the ethanol that they sell, it's 70%. Jacob, can you help us here? What other percent of ethanol you have seen that they sell? Like isopropanol, I know it's 91%. Have you seen any 40 or 50 for ethanol? And what about, well, 100% you never see it.
If the ice seems like
95. So, and again, it can be based on, I guess, the use. Like 95%, yes, you can use it in the skin, but not too frequently. And then, of course, it depends, right? Now, what is the first choice when you have, that's another thing, guys. Very, very important. Open wounds, never ever use alcohol, especially if it's a child. If you want that child to hate you for the rest of your life, put it in there. But you never do that. It is efficacious, yes, but it's not ideal. If you have an open wound, wear your gloves, wash with soap and water first, and then put your disinfectant. In this case, can be hydrogen peroxide or betadine, but never alcohol. Okay. So now if it's something that it's not a scrape, so it's just like for putting an intramuscular injection, it's a small area, yeah, you can use 95%. You can use 70 or 91 of isopropanol. But again, definitely never, even with the lower concentration or whatever you have left from the bottle, never use alcohol. Heavy metals. Metallica, oh. Heavy metals, as its name implies, it's a heavy metal, like argentum, silver, mercury, copper, zinc. Mercury, we don't use it because it is toxic. It can be used combination as mercury chloride. It has a specific uses in pharmaceutical industry, but it is kind of moving away from not using that. But copper, zinc, like copper is an algicide, can be used for cleaning these fish tanks. Silver can be used topical for burn patients. Silver nitrate drops can be used in neonates. Neonates, when they go through the birth canal, if the mom has Neisseria gonorrhea in her vagina, that can go into the eyes of the baby, and that, of course, can create a serious infection in the eyes, leading the baby up to blind. So that in that case, they use silver nitrate drops to prevent what is called ophthalmia neonatorum. And then sometimes copper and silver drops can be used to disinfect water, especially if someone goes to the wilderness. They carry sometimes these little containers with silver drops. And then zinc chloride is found in mouthwash, like Listerine, for instance. The mode of action is denaturing proteins, and it is damaging these proteins by releasing small amounts of antimicrobial activity. That is called oligodynamic action. So here we are looking at a top view of bacteria grown in a petri plate, and then they have two coins that contains silver and this charm of sombrero. So here they move it to the side, and you can see how bacteria didn't grow in this part. And it's not physical interference. How do we know? Well, look at this halo around these coins. So that's, bacteria is not growing around because you have the release of these salts to the surface, and that kills bacteria. Surfactants, soaps. So it's like the nickname for surface active agents. We have acid anionic sanitizers that with the anions react with the plasma membrane, and then they create pores. They are, of course, useful for cleaning our clothing, the eating utensils, and food processing facilities. Quaternary ammonium compounds, they have a positive charge. The cations, of course, are bactericidal, denaturing proteins, disrupting plasma membrane, and they're relatively broad spectrum, except for they're not effective against endospores and mycobacteria. And there is a reason for this. The more structural protection that a microbe have, or in this case endospores, you will have more resistant to disinfectants. Like we saw, for instance, how Pseudomonas aeruginosa wasn't not affected by quaternary ammonium. So here we have an example of quaternary ammonium. This is benzalkonium chloride, so it has this benzalkonium structure with this chloride in here, and then this is ammonium ion, which has a positive charge. So you can test the efficacy of combinations of some disinfectants or preparations. For instance, in this case, you are comparing the efficacy of exposure of these bacteria to these different disinfectants, and you can measure the percentage of surviving bacteria based on seconds. So soap and water, sometimes if you have a lot of bacteria, is not enough to kill bacteria up to 2 minutes. That's why you have to wash really well, a little bit longer than 2 minutes with soap and water your hands. Aqueous Cephuran, it is in its 1 to 1,000 dilution, is not as effective as tincture Cephuran, and then 1% iodide in 70% ethanol, which is tincture of iodide, it is more effective than these other three. So in this case, this is a synergistic effect. Remember, tinctures, it's a mixture of alcohol and another molecule. So those are chemical disinfectants that are used in surfaces. Now, we use as well some chemicals to preserve foods, Like, for instance, sulfur dioxide, it is used to prevent spoilage of wine. Organic acids, it is used to prevent microbe growth in foods, like sorbic acid, benzoic acid, calcium propionate, that can be used in different foods. Inhibit the metabolism of bacteria because most bacteria don't like to grow in acidic environments. And then some of them, they can even give kind of this acidic taste to the food, like potato chips. They have this sorbic acid that tastes in the mouth like acidic. And then we have these two other molecules like nitrites and nitrates. They are important to prevent the growth of endospores from Clostridium botulinum, for instance. They are used primarily in processed foods like meat products, like franks to make hot dogs. And then they're supposed to tell you in the labels whether they contain any of these. Sometimes they can even found in wine, nitrites. So they can be used also to preserve the color of the meat itself, but they're supposed to give you the warning. Why? Because overexposure to these can cause cancer. Some antibiotics can be used as well for preserving foods. And examples will be niacin and natamycin that can be used to prevent spoilage of food like cheese. And then we have these sometimes antibiotics not only used in foods but also in cosmetics, like mascara, etc. And they tell you, if you look in the label, it tells you, well, expiration date. So after that, you're supposed to get rid of it and get new cosmetics. So those were antibiotics and disinfectants, chemical disinfectants that can be used in food products and that you can consume them in a way. Now, these other chemical disinfectants are used for disinfecting lab instruments, medical instruments. Examples will be aldehydes. Aldehydes, they are molecules that can cross-link amino acids, the functional groups of amino acids. That will inactivate the proteins. We have different aldehydes. We have formaldehyde, which is also known as formalin, which is a 30% aqueous solution of formaldehyde. This formaldehyde, it is used as well to preserve specimens. Like, for instance, someone may have a surgery and they have had a tumor removed, or maybe the appendix, they can put that specimen in a jar with formaldehyde and then they bring it to pathology. It is highly irritant and it's having limited use due to cancer risk from exposure. And then we have this secondary effect just by smelling it, like the nose will become runny, your eyes will become teary or watery, and then sometimes the person can start coughing a lot because if it goes into a respiratory system, smelling these gases, it can be irritant. Another example of an aldehyde would be glutaraldehyde. So it's one of the few chemical sterilizing agents that is sporicidal. within 3 to 10 hours. It can be used to sterilize endoscopes and medical and dental equipment that cannot be autoclaved. Like, for instance, these devices to perform laparoscopy, they run it through these glutaraldehyde solutions, the tubes. And then this one may be replaced by a less toxic aldehyde, which is orthopataldehyde, or OPA. Morticians use glutaraldehyde and formalin for embalming. So they withdraw the body fluids and they can inject a specific concentration of this in the bodies. So that's a liquid disinfectant. Now we have chemicals that are gaseous sterilants, and they have a specific use. So what they do as a whole is cause alkylation, replacing hydrogen atoms of a chemical group with a free radical. That's alkylating agents. So we have different gases. We have ethylene oxide, we have chlorine dioxide, and ethylene oxide, it is used in sealed chambers and can be used to sterilize equipment that is large, furniture, like yeah, I think so, right? A mattress is considered furniture. So in hospitals, they use, of course, these mattresses. And then some of these mattresses, they have a cover, of course, that it is mostly not very porous. However, what if a patient vomits? What if a patient has wounds and the blood goes into the mattress? Mattresses are expensive, especially those ones in the hospital. They are reusable. So it is not only disinfecting with the chemical, because these plastics can get damaged. So in this case, they can send it to not manufacturers, like these companies that they deal with these parts, and then they have big chambers. They put several mattresses. They seal the chamber. They inject this ethylene oxide for a particular number of hours, and then they send it back. It is expensive, yes, but of course the hospitals, they might have some extra mattresses there that they can put while they send a batch of mattresses, and that's less expensive than replacing them. And let's say that even that it wasn't that expensive to replace them. I mean, by itself, guys, we, as consumers, regular people, we throw away a bunch of things to the landfill. Imagine if the hospitals will throw away every time the mattresses. We will contaminate more than what we have already contaminated our earth. So it makes sense to reuse. Then chlorine dioxide, it is used in enclosed building areas or water treatment, and can be used also as an aqueous solution for surface disinfection. Any questions so far? Okay, plasma, which is not the plasma that we have in our blood, it is the fourth state of matter. So it is an electrically excited gas that becomes a little bit like a gel. That one also can be used to destroy microbes. So you can create free radicals when you make plasma, and these will sterilize tubular instruments like, again, arthroscopes, laparoscopes. And sometimes this plasma, it is preferred to glutaraldehyde because when you run glutaraldehyde through these tubes, which are sometimes very narrow, I mean, sometimes this glutaraldehyde can dry, and you can try to rinse it, but it make it hard to rinse. And now, you rinse, where does this water with glutaraldehyde will go to? So in this case, plasma, it's a gas that is converted to a kind of semi-solid type of media, and then you run it through these tubes, and then when you just exit it, you put it in a container, it will be converted back to gas. So it's not as bad as glutaraldehyde. It's not as toxic. And then we have supercritical fluids that are similar to plasma, but it is basically in this case is CO2 that is compressed into where you have a gaseous and liquid property. This can be used in the food industry because this can create an acidic environment in which especially microbes like Clostridia won't grow, and can be used as well to treat medical implants like bone, tendons, ligaments that are removed from donors. So more than likely, when someone is going to have a surgery from this type of tissues, it was treated with a supercritical fluid. You can say, well, an alternative can be UV light. But what will UV light will do to this tissue? To the cells in these tissues, like bone, tendon, or ligaments? Cause mutations. You are donating, then in this case, if you would use that, you would donating cancer tissue to the patient, and you don't want to do that. That's why you use these supercritical fluids. And then peroxygens. Peroxygens are oxidizing agents like hydrogen peroxide. Hydrogen peroxide is not good as an antiseptic for open wounds, but it's good disinfectant in non-living surfaces. However, still it's better than something else, like alcohol. And can be used as an aseptic for food packaging. Bioquell, it's a hot gaseous hydrogen peroxide that it is used to sterilize spaces, surfaces such as hospital rooms. And then we have another peroxygen like peracetic acid, like PAA, which is a liquid sterilant, and it is used in disinfection of food processing and medical equipment. So we will stop here.